In this video, we will discuss about photosynthetic pigments. As we know, green plants, they have pigment chlorophyll, and with the help of this pigment, it can do photosynthesis. Photosynthesis is basically manufacture of food. So green plants for photosynthesis require carbon dioxide, water, sunlight, and they require photosynthetic pigments. So if we define photosynthesis, it is the manufacture of food or carbohydrates from carbon dioxide and water in presence of sunlight inside the chlorophyll containing cells. So for photosynthesis, a pigment required cells they are required, that is chlorophyll and other pigments they are required for photosynthesis. So which pigment are involved in photosynthesis, they are called as photosynthetic pigments. Now in this video we'll discuss about pigments. Now which pigments are involved? Chlorophylls, carotenoids, phycobilins, and phycobilins they are mostly found in red and blue-green algae. That we'll discuss in a separate video. In this video we will discuss about in detail about chlorophylls. Now chlorophylls they are the main pigment which are involved in photosynthesis. They give green color to the plant and they are found in plastids called as chloroplast. Now if we see the structure of chloroplast, it is double membrane enveloped, and inside this has thylakoid and grana-like structures, and this is its DNA which is making the chloroplast as semi-autonomous organelle. Now detailed structure of the chloroplast has been discussed in the separate video. Now this I have shown a cell in which chloroplast they are present, and this is the detailed structure of the chloroplast. So in the membrane of the chloroplast, pigments they are found which are called as photosynthetic pigments. Now photosynthetic pigments which are involved in photosynthesis, they can be chlorophyll-a, B, C, D and E. But this chlorophyll-a is the main pigment, or we can say this is the principal pigment which is involved in the photosynthesis. This is found in all the plant, so this is also called as universal pigment. Now chlorophyll-a can be found with chlorophyll-b. When the combination is chlorophyll-a and chlorophyll-b, then this is found in plastids called as chloroplasts. Now but chlorophyll-a may be found along with chlorophyll-c. This may be found with D, this may be found with E. When the combination is chlorophyll-a and chlorophyll-c, this is usually found in brown algae, diatoms and dinoflagellates. And when this is found along with red algae, this chlorophyll-d, this is found in red algae. When this is found in with chlorophyll-e, this is found in yellow-green algae. And the plastids in which these combination of pigments are found, they are found in chromoplasts. And if chlorophyll-a and chlorophyll-b is there, now this will be found in chloroplasts. And if chlorophyll-a is present along with C, D and E, this will be found in the chromoplasts. Now we'll discuss in detail about structure of chlorophyll-a molecule. Now this was given by Willstater, Stroll and Fischer. And if we see its formula, this is C55 H72 O5 N4 and magnesium, and color is bluish green in color, and this is found in plastid chloroplast. We can extract with the help of different organic solvent like petroleum ether, ethyl ether, many other organic solvent can help in extraction of the chlorophyll molecule. Now this is found in plastids chloroplast as we discussed. Now chlorophyll-b has a slightly different formula. This has C55 H70 O6 N4 and magnesium ion. Now we'll discuss in detail about structure of chlorophyll-a molecule. Now if we see structure of chlorophyll-a molecule, it has 4 pyrrole rings, 1, 2, 3 and 4. Each pyrrole ring is made up of 5 atoms, that is 1, 2, 3 and 4 carbon and 1 nitrogen atom. Now each pyrrole ring will be having again 1, 2, 3 and 4 and 1 nitrogen atom. So there are 1, 2, 3 and 4 pyrrole rings. Now we can see here at one position methyl group is present, at 1, 3rd, then 5 and 8. Methyl group is present. What is methyl group? CH3. Now if we see here at 2 position, at this position vinyl group is present, CH, CH2. At 4th position here, ethyl group is present. Now these 4 pyrrole rings, 1, 2, 3 and 4, they are linked with each other by magnesium ion. Now magnesium ion is connected with this 4 pyrrole ring by 2 covalent and 2 coordinate bonds. And these 4 pyrrole rings are also connected by these bridges that is CH, like these are called as methane bridges. So these are one pyrrole ring, this is another pyrrole ring. Now this is connected by this bridge. Now this pyrrole ring and this pyrrole ring is connected by this bridge. So these are called as methane bridges. So this is forming a head. This is called as porphyrin head. And we can see here also, this is also tetrapyrrolic in nature, mean 4 pyrrole rings are there. So this head is called as porphyrin, right? And another thing which we can see here is it also has a fifth ring, right? Now this is the fifth ring here. This is called as cyclopentanone, and this is attached with the third pyrrole ring, right? Is it clear? Now we can see here at the 7th position, this is the propionic acid, and propionic acid is attached to the long phytol tail. This is the long phytol tail which is attached with the propionic, and this is present at the 7th carbon. Here we can see here, this is the 7th carbon where propionic acid is attached, and to this is attached a long tail. This is called as phytol tail. So we can 4th pyrrole ring is esterified with the long chain alcohol called as pyrrole ring, right? So this is about structure of the chlorophyll A molecule. Now if we see the structure of chlorophyll B molecule, it is the same. Only difference is chlorophyll B at 3rd position here, this will have aldehyde group, CHO group. So we can also call chlorophyll B is aldehyde of chlorophyll A molecule. That's why its empirical formula is C55 at 70 O6 N4 and magnesium ions. So this is about structure of the chlorophyll A molecule. Now, as we discussed, we can extract the chlorophylls with the help of different organic solvent, petroleum ether, ethyl ether, acetone. So when we extract the chlorophyll molecule, that form a solution called as chlorophyll solution. So when we filter it, centrifugate it, and we can measure its absorption spectra with the help of spectrophotometer. So what is absorption spectra? That is absorption of light by the chlorophylls. An instrument which we are using, this is the spectrophotometer, and this will work at different wavelengths of light. So the portions of the light spectrum absorbed by chlorophyll solution which appear as dark band constitutes the absorption spectrum. So here we will discuss how this graph is created. So we can see here, if we take chlorophyll A bands, right? So we can see here maximum peak is here and maximum peak is here. So this is maximum peak is in the blue violet zone and in the red zone. So this is chlorophyll A is absorbing at in the blue violet zone 430 nanometer and at red zone 662 nanometer. And chlorophyll B will be absorbing at 455 and chlorophyll B will be absorbing at 644 in the red region of the spectrum. So this is the band of the chlorophyll A molecule and this is the band of the chlorophyll B molecule. So maximum peaks they are found in the blue and the violet region of the spectrum, and this mid region is flat. If we see, this is about absorption spectra of the chlorophyll A and chlorophyll B molecules when we extract with the help of organic solvent, and we can measure with spectrophotometer. We can also see the effectiveness of this wavelength, like where photosynthesis is maximum. So it was seen that maximum peak is found in the blue zone and red zone, right? So maximum photosynthesis is also found in the blue-violet region as well as in the red region. So this is called as action spectra of chlorophyll A molecule. So here this is absorption spectra of chlorophyll A and chlorophyll B. Here this is the action spectra of chlorophyll A molecule and this is the absorption spectra of the chlorophyll A molecule. So we can say what is absorption spectrum? The portion of the light spectrum absorbed by chlorophyll solution which appear as dark band, this constitutes the absorption spectrum, and how much these wavelengths they are effective and causing the photosynthesis, that is the effectiveness of different wavelength of light on the photosynthetic activity of an autotrophic plant bean which can manufacture its own food, which is green in color, is called as action spectrum. So this is about absorption spectra and action spectra. Like we discussed, photosynthetic pigments, they are chlorophylls, carotenoids and phycobilins. And in chlorophylls, chlorophyll A is the major pigment or principal pigment which is involved in the photosynthesis. So chlorophyll A may be found along with chlorophyll B, this may be found with C, this may be found with D, this may be found in with E, but A is the major pigment involved in the photosynthesis, and this is found in chlorophyll A and chlorophyll B, they are found in plastid chloroplast, and when this is found with other chlorophylls, this is found in the chromoplast, and maximum absorption of the light occur in the blue violet zone as well as in the red region of the spectrum, and maximum photosynthesis will occur in the blue violet zone and red region of the spectrum. So this is all about chlorophylls. If you have any questions, you can ask me in the comment box. Thank you for watching my video. If you like my video, please like, share and subscribe. In the next video, we will discuss about carotenoids.